has just left and is sitting here, and another, if he's not enjoying lunch, will be coming soon. Anyhow, this is the map of the world, and this is the interaction, and I assume most of you can't actually see the most important bit because somebody's head's in the way, but this is easily fixed. So now we've got Argentina in the proper place at the top of the world. I'm afraid I can't do that for the rest of my slides, so you'll just have to jump around and try to see what's at the bottom. So what I want to talk about is budgets. Not financial budgets, but carbon budgets. So plants grow by taking light, carbon dioxide, nutrients, and water out over their leaves and their roots, and out of they make more leaves, more roots, then they get more light, more carbon dioxide, more nutrients, more water, and they go on growing. And the questions we're interested in is how do they gauge how much resources they've got, and how do they use this information to decide when and how they're used for growth. The budgetary problem that plants have is in the day they have light, and in the night they have no light. That's when they get all their carbon. That's when, and here, from this point on, they've basically got to survive the night. They can't go out to McDonald's. They can't go out and have a tapper. They basically have to make sure they have enough stored here to last the night. And many plants, they do this as starch, where in Arabidopsis up to 40% of the carbon is accumulated during the day and is broken down in the night. This is pretty well linear, and it pretty well runs out. So this is optimized for growth. It lasts until the end of the night, so they avoid transient starvation, but it's almost all gone. That means they're investing immediately in growth. If they can't make starch, they can't grow in a light-dark cycle. That's been known for about 25 years. So we started looking at this using omics. So we were interested in a lot of complicated changes which are going on in these whole plants. Many things are changing simultaneously. So we used a whole series of simple experiments where we altered sugars, we altered light, we looked at what the clock was doing, and we then used this information to interpret a lot of omics data for what was going on during diurnal cycles. I'm going to summarize this work in one slide. During the night already, hundreds of genes for protein synthesis and growth are being repressed. But what happens if you extend the night? Within 2 to 4 hours, hundreds of genes for catabolism are induced, growth stops within 2 to 4 hours, polysomes are dissociating, metabolism is changing, and though the plant is just living on the nice life edge, it has about 1 hour of carbon left. So it has to get this right, and if it does get into a fix, it's like getting into the red with your banker. There's a downside, and here, after you illuminate, there's a delay until growth starts again. So plants have to stop running out of their carbon. It's been known for a long time that if you grow plants in different conditions, for example, in a short day, they actually make starch faster, they break it down more slowly, and this is linked with a decrease in the rate of growth. So here we have what is a system response, which is linking changes in metabolism with growth, and this is what we're interested in. Or putting this more simply, this is what the plants manage, which you call a balanced budget, and this is what a lot of us do. This is what most of our politicians do and a lot of our bankers. And I won't go into more detail. Some are further down than others. So that's the issue. Now, plants not only have to deal with long-term changes, actually, at least where I grew up, there wasn't climate, there was weather every day. So a plant, there are short-term, unpredictable changes. You never knew in advance how much light you were going to have, how much photosynthesis you could carry out. This meant you'd be starting the night with different amount of starch, and you never knew what conditions were going to be like in the night. It might be warm, it might be cold. So you're actually having to solve, the plant will have to solve these problems continually, what I would call on the hoof. It has the long-term problem and the short-term problem of staying out of a credit freeze. So, this is the key experiment. If you've understood this experiment, you can go home. This was done by Alex Graff in Alison Smith's lab. So he grew colonol in a 12-12 cycle, and then suddenly one day, without telling it, no foreadvance, darkened it 4 hours too early. And what did the plant do? It immediately decreased the rate of starch breakdown. And it broke it down so it just lasted to the end of the night. So how the hell do these plants know how long the night was? Well, the answer, of course, is the clock. And these are 2 clock mutants. I won't go into the details of what sort of mutants they are. Here's a mutant which actually runs on a 20-hour period, and its starch runs out at about 19 to 20 hours. Here's a mutant which is running on a 28-hour period. This is like my son. Yeah, he never gets up till 1 o'clock in the morning, and in the afternoon. And the starch a lot of it is still left at the end of the night. So this links it to the clock. Now this is an experiment to see if you're thinking. So this is a question. We grew plants in a 14-14-hour cycle. Not a 12-12 cycle, but 14-14. So they had a 28-hour day. What a lot of us had yesterday. But that's every day of their life. My question is, when does the starch run out? Who thinks it ran out at 24 hours? Hands up. Who thinks it ran out at 28 hours when actually they were having their dawn? Who doesn't know? And who couldn't care less? Okay, the clue, which is hidden, is the clock always operates with a 24-hour period, because that's the way it's been for the last millions of years. So clock genes peak at 24 hours, even if you're growing them in a 28-hour cycle, and the starch also ran out at 22 to 24 hours. And that was the second piece of evidence in the paper we published with Alison Smith, that the starch regulation is regulated by the clock. So now I want to turn to starch synthesis. And what is the clock doing? And I won't give you a final answer. So starch is broken down in a very complex way. First, it's a granule, it's crystalline. And first, the surface is modified by phosphorylating and dephosphorylating to solubilize up the surface. It's then degraded by a series of amylases, which release mainly maltose, which then goes out into the cytosol and is turned to sucrose. So what happens when we suddenly, maltose is important, it goes up when starch is being degraded. What happens when we suddenly, when we suddenly have an early dusk, this is what would have happened, and this is the maltose levels when we have a suddenly early dusk. So the answer is the degradation is being stopped at a very early step. This is not a backup from the plant. This is an inhibition of the early steps in starch synthesis. This is another degradation. This is another line of evidence that it attacks the surface of the starch grain. We took advantage of mutants, which are actually needed to make the starch grain. They sort of trim things around. And if you take the gene, and if you take the mutant, it accumulates half of its carbon as phytoglycogen, like you have in your liver if you've had something to eat today. And the rest is starch. And when we look at this, the phytoglycogen is degraded quickly in an unregulated way at the beginning of the night, and the starch is degraded in a linear manner over the entire night. So we need features of the starch grain surface to have this regulated breakdown. Now this experiment has another very interesting finding. These experiments were done with plants growing at different night temperatures. And what you'll notice is the state of starch breakdown is completely temperature compensated. And that's amazing for a biochemist. I would expect it to be inhibited 2 to 3-fold. It's temperature compensated. Now this is an experiment where we had the plants growing in a 24-24 cycle and then suddenly decreased the temperature to 12 degrees. Exactly the same thing, the same rate of starch breakdown. So our idea here is that there's basically a break on the starch breakdown, and it's being tied to this rate, which will get it to run out at the end of the night, irrespective of the temperature. And you won't see it at the back, but exactly the same thing happens if you grow them in a cold night and then suddenly warm it up at the beginning of night. Rate of starch breakdown is independent of the temperature. So this leads to the following concept, that we have the clock putting a brake on the rate of starch breakdown to make it last to the end of the night, not too fast, not too slow. This raises a whole series of questions which I'm not going to answer. How do you measure the amount of starch? I've got some really crazy ideas, so if there's an Argentinian who wants to join the lab to drink a lot of beer and test these ideas, they're very welcome. How do they measure the time to dawn? I don't think we can do it with a clock as we now know it. How are these cues integrated, and what biochemical mechanism is then doing things? And I'm going to come on to that. But first, I want to briefly go on to feedback regulation. Actually, I think I'm going to skip all over this to save some time. I'll just run through this quickly. We don't need it, so that we actually save some time. The slides I've jumped over, because I don't think I need them for the message today, is that sucrose feeds back via a signal metabolite called trehalose 6-phosphate to inhibit starch breakdown. And this is interacting with the clock. And this is basically a system that if sucrose is being used for growth, then this feedback regulation is removed, and we stimulate starch breakdown, and the clock is then putting a brake on it and keeping it at the rate that it should be. So I've just jumped over 3 or 4 minutes of slides. Now, this brings up a whole series of questions about the mechanism. We're approaching this with forward genetics, and by Alison Smith is looking for mutants with premature starch breakdown. We're exploring this loop which we found here, and we're looking for suppressor mutants of this loop. We're looking for mutants which have lost the temperature compensation. We're doing biochemical genetics, and this is together with Alison Smith in Norwich and Sam Zeman in Zurich. I don't want to go into any of this data and just point what it looks as if we're looking at complexes which are being built up at the starch grain surface and which are somehow regulating the rate. Now, this is going to be another 2 or 3 years' work, I think, to sort it all out, but I now want to move on to the other side of the coin, the use of the carbon for growth. So it's being exported and used for growth. So I'm going to talk now about a series of experiments where we're looking at the role of the clock in regulating growth using roots as a system. So this, Joachim Fizan set up a system which allowed very good temporal and spatial measurement of the rate of root extension. And this shows what happens in a light-dark cycle and then in a free-running cycle. You can see it's oscillating, so we have circadian regulation. You can also see the rate is rising. This has become, we've gone from a light-dark cycle to continuous light. They've got more carbon, so that tells us already rate of growth is regulated by the amount of carbon, it's regulated by the clock, but within the 24-hour cycle, we can see growth is already rising at the end of the night, so they've obviously got reserves there. This picture alone tells us a lot. Now, this is a more detailed experiment with 3 different photoperiods, a short photoperiod, 12 hours and 16 hours light. There are light-dependent changes which I'm going to ignore for the talk. I just want to focus on the circadian changes. They decrease growth in the first part of the 24-hour cycle, and it recovers in the later part of the cycle. So what we can see here is really graded growth, and very important result. The overall rate of growth is really linked to the amount of photosynthesis, but when it's growing and the timing is changing through the cycle, and in what I think is a conservative way, it's saving carbon towards the end of the night, and I see this as mainly saving in case something goes wrong. So I always tell my son, go out and buy your next piece of apparatus for your guitar at the end of the month, and not at the beginning, because you don't know what else is going to happen before you get your next lot of pocket money. So now I want to go into what the clock is doing here. It's doing 2 things. One is it's regulating the starch turnover to make sure that there's enough carbon through the entire 24 hours. And this we can see, for example, in this double mutant, which I showed at the beginning, this mutant which the starch is broken down too early, runs out at 20 hours. We see an inhibition of growth at the end of the night when the starch runs out. And this can be recovered just by giving sugars. So here we have disturbance of a metabolic process in the starch turnover leading to no starch. We see an inhibition of growth at that time, and we can complement it biochemically. So I think this shows very clearly the clock is doing things here, and the clock is doing something which is important for growth. The second is the clock, however, is also responsible for this depression of growth in the day and then letting growth run towards the end of the 24 hours. Now we can observe that. And then I'm going to give one example of a mutant where we can disturb it. This is a mutant called L3. This is one of about 100 or 200 early flowering mutants in plants. Not particularly interesting, actually, for that reason. But this plant, this actually grows faster in the day, and its growth starts to drop during the night. This is the very bright blue line. So it's faster in the day, and it slows down at night. So it's completely flipped. I'm not going to go into a lot of detail. It shows exactly the same response in the free-running cycle. This mutant is unable to depress growth in what should be the light period. I want to point out, the only thing I want to point out is this mutant has very recently been shown by a couple of the leading clock labs to be a really integral part of the core clock gene. It's part of the so-called evening complex, which is sort of sitting here. We're just starting to find out about what it does, and a very nice paper from Steve Kay's lab has linked it with a regulation of sort of seedling shoot growth by light, Steve Kay, and so we can start looking at genes here which may be also linking the clock to vegetative growth in earlier plants, and I don't want to go into this in more detail. We also have a lot of candidates out of omics study across photoperiods. I want to now switch to the last part of the talk. Yep. About another 5 slides. You may not even have to get up. Now, this isn't really growth that we were looking at. This was things getting bigger. It's rather like a balloon being blown up. Most of a plant is vacuole. It's water. And when you actually look physically at a plant growing, what you're looking at is the vacuoles getting bigger. we really need to look at growth as well. And this means that the synthesis of cellular components. And we're taking two approaches to look at this. And I think I'll only go over one of them in detail today. One of them is actually simple, old, whole plant physiology and a little bit of maths. So we measure photosynthesis in the day, we measure respiration at night, and we measure a lot of metabolites, and we add them all up. And in fact, it's very easy because 80% of the carbon which accumulates in a plant is in starch. So we just ask what accumulates in a plant between dawn and dusk. So those are three things: photosynthesis, respiration, and how much carbon is piling up in the plant. And then we can calculate growth in the daytime is photosynthesis minus the stuff that piles up in the plant, the starch and other small molecular weight metabolites. And growth at night is basically the starch which is mobilized minus respiration. It's very, very simple. And I want to show a couple of results from this. This is photoperiods. So this is how growth is distributed between the night and the light in different photoperiods. In short photoperiods, almost all the growth is occurring at night. This is almost counterintuitive. And the longer the photoperiod becomes, the more day there is. Now, this is growth in the entire photoperiod, but here, jammed again. Well, down here, I would have calculated them on an hour basis. I think the train going by has stopped it. Actually, what I'm going to, it's completely crashed. Okay. Okay, now we're back. This is calculated on an hour basis, and it's still the case. This is a second experiment where we change the night temperature. And you can see that the rate of growth actually goes up when the night temperature decreases. And remember, the starch turnover was also temperature compensated. Plant growth in these conditions is completely temperature compensated for growth at night. The same thing actually happens if you have them cold all the time. Oh, yeah, this was all the time. Sorry, that last was all the time, and this is only cold in the night. And they're also temperature compensated. Now, I don't want to go into the details on that. Yep, I'll make it. I'm going to put out a concept. Plants have a predictive and unpredictable, fluctuating, unpredictable environment. This means they're always going to have different rates of photosynthesis, also respiration because of temperature. Their starch turnover is, over a wide range of conditions, a highly conserved response. It goes up in the day and goes down at night, and it doesn't run out before the end of the night. I see this as basically a system response which the clock is setting. The growth, the rate of growth in the day or the night, the plant's completely opportunistic. Sometimes it grows more in the day, sometimes it grows more in the night. The main thing is that it does not disturb what you could call the finance minister. And the finance minister is the clock setting starch turnover. Or any one day, it doesn't matter how fast or when you grow, as long as you don't get into the red. So that's my take-home message about how clocks do better than our politicians. Now, we've left all sorts of questions about the mechanisms. I deliberately left them out so I could get done in 20 minutes. So on the last slide, I'll just point out what we're doing. We're extremely interested in getting into individual cellular growth processes. We're looking at protein synthesis as an example because it's a major growth process and